microbiology, immunology, and tropical medicine. David is the clinical director of the GW Vaccine Research Unit. And I, sort of gave him an impossible task a few weeks ago. And I only gave him a few weeks' notice. I think there's a lot of interest in us learning about vaccines, and there seems to be a lot of different companies out there looking at different types of vaccines. And the questions are, what are the targets? What are the likely successes? What are the impediments? And when can we maybe expect a vaccine? So it's a difficult task that I asked David to do, and he was gracious enough to do it. So David, thanks for being here again, and take it away.
Thanks so much, Dr. Wasserman, for that introduction. It's true, I think it was about a month ago that you first asked me to give a presentation on COVID vaccine development, and I said, well, I don't think we have enough results. There haven't been any published data yet on any clinical trials, but man, what difference a month makes. We have results of 2 phase 1 clinical trials already, and about over a dozen of different candidates are in clinical trials. So I have actually more than I thought that I'd be able to present. Get this working. These are my disclosures, which unfortunately are none, except perhaps that even after living in BC for almost 2 decades, I still remain at heart a Canucks fan. I'm going to answer one of your questions, Dr. Wasserman, very quickly, and that's what are the targets? Well, there's one, really, in terms of what the immune response to an effective vaccine is being targeted towards, and that's the spike protein. So I'm not going to go anything into the permanent disease that's been covered very well in the past several months by Dr. Axelrod and others. But I just wanted to remind you of the structure of the virus. So it's an RNA virus. It does have an envelope into which several glycoproteins are inserted. The most important in terms of pathogenesis is the spike protein, which is a trimeric protein that is responsible for entry of the virus into human cells. And it does this by the spike protein binding to the ACE2 receptor on the surface, at least initially on the respiratory epithelium. When it does bind to ACE2 receptor on the surface of cells, it does undergo conformation, and it's important to point out because it does have implications in vaccine development. Because all of the vaccines currently in development are modeled on the pre-fusion, so before it actually binds to the ACE2 receptor conformation. Once it does bind, and the part of the spike protein that binds is called the RBD, or receptor binding domain, under the RBD domain undergoes a conformation. They call it standing up. That's quite significant and is essential for the actual mechanism of cell entry, which is a form of endocytosis. And once, of course, the virus enters the cell, then the RNA within is released, and you get production of viral proteins. Remember that difference in conformation between the pre-fusion and the fusion forms of the spike protein. So what we do see in terms of immune response, I won't go over this in detail, in those who are exposed and infected with the virus. This just shows that, at least to the receptor binding domain, when you're looking at binding antibodies simply by ELISA, there are IgG, IgM, and IgA antibodies that are induced compared to negatives who have not been infected or exposed to the COVID-19 virus, SARS-CoV-2 virus. And in addition to a humoral immune response, there is also a cellular immune response. So this just shows the CD4 T cell response, both to spike and non-spike proteins of the virus. And this is very encouraging for vaccine development because we want to see a strong CD4 T helper cell response that will be responsible for induction of immune memory, so that if we can mimic that with a vaccine modeled on the spike protein, that we could have long-lasting immunity. Interestingly, and apologies for some of my slides, the conversion from Mac to PC doesn't always work so well. But interestingly, when you look at people who have not been infected with the SARS-CoV-2 virus, and these are just taking samples that are stored in the freezer from before the pandemic, so we know they're negative. They do, although they don't have an antibody response, don't have detectable antibodies to the SARS-CoV-2 virus, you can detect cellular immune responses, so CD4 responses to mainly non-spike proteins. So that's here on the right, you can see, versus the spike protein. But that is interesting and something that we're definitely going to have to monitor with, during clinical development of the vaccine, because it could potentially, and we don't have answers to these questions yet, in terms of at least does that pre-existing immunity, which is thought to relate to prior infection with the common circulating cold coronaviruses, it could modulate severity of disease with infection. That's something that's obviously being looked at now, but in terms of vaccine development, could also potentially modulate the response to vaccination and could potentially confound outcomes of vaccine trials. I thought that was important, so I highlighted it. So why spike? Well, there is data from the SARS-CoV-1 virus from almost 2 decades ago that monoclonal antibodies, at least, this was just an animal experiment. It never made it to human trials because obviously that virus disappeared. But in a ferret animal model where ferrets were given monoclonal antibodies specific to the SARS-CoV-1 spike protein and then challenged, those who on the right here who got the monoclonal antibody were protected compared to controls. So that's one piece of evidence, but there is other evidence that we think that the predominant target for vaccine development should be the spike protein. So where are we in terms of development of a vaccine? Well, as of this week, and this is pulled from 3 days ago, I checked this morning and it hasn't changed. So there are 13 different candidates currently in clinical development, so have been given to at least one human. The ones at the top are more advanced and further along in terms of vaccine development. In the US, the first one that went into clinical trials, and which I'll talk more about, is the Moderna mRNA vaccine. The University of Oxford AstraZeneca vaccine is going to be the second one that's going to go into a phase 3 trial, according to current plans, at least in the US. The Chinese have a bunch of different constructs. Not as much is known about those, and they're going to have obviously difficulty doing efficacy trials given their current incidence at least in China. There are a bunch of different platforms that are being tested. Some are traditional ones, such as recombinant proteins, the Novavax vaccine with an adjuvant, simple inactivation of the virus, like growing the SARS-CoV-2 virus up in a cell culture, inactivating it and using that as a vaccine. That's being done in China. And then a lot of newer technologies. So mRNA, we don't have any licensed vaccine that works based on this technology. It's fairly new. DNA, adenovirus, vector vaccines, and so on. So I'll talk a bit more in detail about some of these. There's a lot more besides those 13 currently in clinical trials that are going to come online in terms of clinical development before the end of this year. It's estimated there will be over 60 vaccine candidates in clinical trials. And this is happening all over the world. Concentration of development activities in the US, Europe, and in China, but lots of other spots, including Brazil, Asia, Australia. And also as of this week, there's about 160 candidates, aside from those 13 already in clinical trials, that are at some stage of preclinical evaluation. And this is almost changing by the day. So this obviously has been an unbelievable paradigm shift in terms of the timeline for clinical development. So up at the top is our traditional paradigm, phases done in sequence, looking at results of the phase 1 before you go into phase 2, finishing that, including long-term follow-up before going into phase 2B, and then finally phase 3 and licensure. This has been all compressed. So it boasts the target selection, preclinical evaluation, toxicology testing, is all happening concurrent to initiation of clinical trials, and even the stages of clinical development are overlapping, with phase 2 and phase 3 starting even before the phase 1 long-term follow-up is finished. So I've talked to a lot of people, not necessarily in the vaccine development space, but what they think about this, and a lot of them say that they think it's very risky. This is kind of a summary of a talk that Tony Fauci gave, I think last week at the Keystone Symposium on COVID vaccine development. According to him, his take on things, and I highly respect him, he's one of the giants of public health, that the main risk is really financial, and that speeding up this development has just meant spending a lot of money much quicker to get things done, including producing all the vaccine we might need even before we know it is effective at all. His take on the risk in terms of safety is that there is not any compromise in safety that's taking place. I'm not sure I 100% agree with that statement, and I have a few slides that go into a little bit more detail on that. But it is true that we're still basically following the same scientific progression of studies. They're just overlapping, but they are feeding one into the other. The design is the same as if we would do this in a non-pandemic situation. It is true that what he said about increasing the safety consideration and safety evaluation in phase 3 trials, as you'll see in a minute, we're going to start a phase 3 trial here of the Moderna vaccine in July, and we are doing a lot more safety evaluations than we would otherwise in a phase 3 trial. I'm sure you've heard the term warp speed. I have another slide on this in a little bit, but this is definitely moving at very fast pace as opposed to usual traditional vaccine development. And I'm sure a lot of you have heard some of these potential concerns thrown around for a SARS-CoV-2 vaccine. I personally don't think that there's much risk of these, but for some reason, all of the publications are repeating this. The first is antibody-dependent enhancement of disease. So that's if someone's vaccinated and then gets exposed to the virus, that the actual infection and disease caused by the infection is worse than if they were not vaccinated. This is something this was presented, I think, last week regarding dengue. This is something we see with flaviviruses, particularly dengue, where there's more than one serotype and you can be infected sequentially with more than one serotype, and it's caused by FC portion mediated enhancement of infection into phagocytic cells such as monocytes. There is a coronavirus where this is seen, a cat infectious peritonitis virus, which is a coronavirus. I think this is a lot different than SARS-CoV-2 because the target cell for infection of this particular coronavirus is actually macrophages, which is not a target of the SARS-CoV-2 virus. So we don't have any evidence of this to date. There have been animal studies at least done with SARS-CoV-1 that did not demonstrate ABE. So I personally don't think that this is going to be a big risk, but obviously will be monitored for. And then something also called vaccine-associated enhanced respiratory disease. This was seen with old vaccines for measles and RSV, whole cell inactivated vaccines. And this was really due to an induction of a high ratio of binding non-neutralizing antibody to neutralizing antibody. So basically, induction of antibody that did not protect or neutralize the virus, but that was able to stimulate or be associated with immune complex deposition, formation and deposition, and complement activation. So it's, again, something that if we have a vaccine that induces high levels of neutralizing antibody, we hopefully should not even have to worry about this. One potential other concern, and Dr. Axelrod did touch on this, is that at least the serology data that we have to date on infection with SARS-CoV-2, this is data from China, not a lot of people, but looking at time course of IgG titers. So in symptomatic versus asymptomatic infection and symptom, this was based on PCR, the actual diagnosis. So 8 weeks after the initial blood draw, they did see a quite significant reduction in IgG levels, median IgG, about 70% in both symptomatic and asymptomatic. So hopefully we will have a vaccine that doesn't have the same drop-off in IgG levels, but that is concerning both for possible, post-infection reinfection because of waning immunity, but it's obviously a kind of concern also for vaccine development. So the first clinical trial that's been reported in the literature was a phase 1 trial done out of China. This was a recombinant replication deficient adenovirus type 5 vector vaccine that expressed full-length spike protein. This was a dose escalation trial, single injection. About a month after the single injection, they did see induction of neutralizing antibodies. We don't know what level, obviously, we're going to need to see to have protection, but they did, and 75% at least of the high dose group have neutralizing antibodies. What I found interesting about this paper is that at 5, a lot of adults, usually over 50%, had existing antibodies to this vector because of prior infection, which we know from other vaccine constructs could interfere with response to vaccination. They didn't report difference in antibody level between those who had baseline pre-existing Ad5 antibodies versus those who didn't. They did look at T cell responses and reported they were lower in those who were seropositive at baseline for Ad5. What about what's going on here in the US? Operation Warp Speed was launched, I think, in May, so about a month ago. Not just focused on vaccine development, but this is a White House-driven initiative with a lot of different partners in the government and private firms. I'm sure you've heard about this, looking at developing new vaccines, therapeutics and diagnostics. There's also a group that's been put together by the NIH, led by Frank Collins. Tony Fauci's on this as well. I actually have to say it's quite confusing to me, even someone who's quite involved in vaccine development, what all of these different partnerships are doing and consortiums, because they seem to be looking at coordinating the same thing as the Warp Speed group does. I guess that remains to be seen. But in terms of vaccines, what has been chosen, at least by BARDA, NIAID, HHS, for investment are these 5. So the first 2 are already in clinical trials, as you'll see in a second. The next 3 are still in preclinical, but hopefully by the end of this year, will all be in clinical trials as well. And they all have different platforms that they're looking at. So I guess they decided to choose one from each different platform and based on where they are in terms of clinical development as well. So the first one is the Moderna vaccine. This dates back to March, when in record speed, really, they went from sequencing the genome to having a vaccine that went into the first human in mid-March. I'll give a little bit more data about this trial in a second. But the idea behind an mRNA vaccine, and again, we do not have any licensed vaccines that use this technology, quite new. But the idea is that you have mRNA that encodes for the protein of interest, in this case, the full-length spike protein, that's injected intramuscularly, gets into muscle cells where it can then use the cellular machinery to produce the protein of interest, to which the body will then develop an immune response, hopefully protected. The Moderna construct called mRNA 1273 is the mRNA is not just injected naked. It does have to be encapsulated, in most cases, a lipid nanoparticle. In Moderna's case, that nanoparticle consists of 4 different lipids, 1 proprietary, 3 commercially available. And the mRNA enclosed in the nanoparticle encodes for the pre-fusion stabilized spike protein that does have 2 amino acid modifications that allow it to stay in the pre-fusion conformation, so it doesn't automatically go into the post-fusion one. So I found this figure quite helpful in understanding how this all happens. So the mRNA LNP that's injected intramuscularly, the lipid also, besides providing stability and protecting the mRNA, also facilitates its entry by endocytosis into cells such as myocytes, where the mRNA is then transcribed by cellular machinery into the S protein that can then be secreted or expressed on the surface of the myocyte. And then can be presented to the immune system, including antigen-presenting cells such as dendritic cells that can migrate to the draining lymph nodes and interact with naive EMT cells to produce both a cellular and a humoral immune response and produce, in the end, neutralizing antibodies, we hope. Interestingly, not only can the LNPs deliver the mRNA to myocytes, they can also deliver it to antigen-presenting cells directly. And then the antigens can be processed, the spike protein can be processed intracellularly and then expressed by the APC to, for example, CD4 T cells, also to end up producing, in the end, a neutralizing antibody. So the timeline for the development of the Moderna vaccine has been very rapid, as I mentioned. The first human injected on March the 16th. We had the first positive interim results reported in mid-May. The first trial was done under an investigational new drug application by the NIH because they sponsored this first trial. It was done in Seattle initially. And then the company itself submitted another IND for the same vaccine in the end of April for the phase 2 and 3 trials. They've already contracted with the manufacturer to ramp up production, and phase 2 trial was started at the end of May. So I do have some initial results from the phase 1 trial that I'm going to present. This is supposed to be published in New England tomorrow. So I wasn't told I have to keep this confidential, but please don't copy it and send it out to the press. So this was a traditional phase 1 trial looking at safety, immune response, and reactogenicity, dose escalation, looking at 3 different doses, 25, 100, and 250 micrograms, 2 injections intramuscularly spaced a month apart. The first part of this trial enrolled 45 individuals between the ages of 18 and 55. That was what was done in Seattle at Kaiser Permanente. They have expanded it to a site in Atlanta, where they have now injected older individuals on 2 cohorts, 56 to 70 years and over the age of 70. The data I'm going to present is all in the 18 to 55-year-olds because they were done first. And they have amended the protocol, phase 1 protocol, to add a 50 microgram dose as well across all 3 cohorts. In terms of safety, it looked like it was well tolerated. In the 25 and 100 microgram cohorts, there was only 1, and that was 30 volunteers. There was a single severe grade 3 adverse event in the 100 microgram dose level, and this consisted of a transient injection site erythema, just based on measurement, over 10 centimeters in diameter. And that was after the second injection in that participant. And then in the 250 microgram cohort, they did have 3 out of the 15 who had grade 3 systemic symptoms, fever, chills, headache, myalgia, fatigue. 1 of the volunteers actually ended up going to urgent care because of this. And it was after the second dose again, ended up being sent home and then became lightheaded, vomited, had an episode of syncope, but then recovered by the next day. No serious adverse events. All adverse events have been transient and self-resolving. This shows the binding antibody levels, and you may have seen the initial antibody levels presented in a press release, which were only on 8 subjects, I believe. This is all the full cohort of 45 individuals. At the 3 dose cohorts, you do see a dose response in terms of better responses after the second injection in all 3 dose levels. There didn't seem to be much of a difference between the 100 microgram and 250 microgram doses in terms of the binding antibody response. But in comparison to convalescent, so people who recovered and their antibody levels, these are IgG responses, the vaccine did induce higher antibody levels at both 100 and 250. That was what I just showed was to the full-length spike protein. This is just to the receptor binding domain, similar results. And most interestingly are the neutralizing antibody response, both using a pseudovirus and then live SARS-CoV-2 virus. For the live virus assay, they so far have only done 2 time points and only in the 25 and 100 microgram dose groups, but good neutralizing antibody responses after the second dose of vaccine and higher than the few that they've already looked at in terms of the convalescent serum. And also good responses with the pseudovirus assay. And much better than you saw with the convalescent. But that's only 2 weeks after the second dose. So, you know, concern about longevity of the immune response. So we don't have any data yet in the phase 1 because there's not been enough time to look at it. This is from a different construct. So this is influenza using, again, from Moderna, the same platform, mRNA platform, looking at 6 months post 2 doses of the flu vaccine that they studied, and did even 6 months later still see significant IgG antibody responses. So hopefully we'll see the same thing with the SARS-CoV-2 vaccine. Phase 2 trial has already been started for this construct. First volunteers were immunized on May 29th. This is just looking at the 50 and 100 microgram dose groups. They've dropped the 250, presumably because of reactogenicity and the lack of an improvement in the antibody response going up that high. It's 2 dose groups compared to placebo, 1-to-1-to-1 randomization, 600 volunteers. I think they've all been vaccinated. They've all received their first dose of vaccine by now. And we don't have any results yet. So phase 1 enrollment complete. I just showed you initial results that hopefully are going to be published within the next few days. Phase 2 underway, and the plan is to start the phase 3 in July, as I'll show you in a bit. The phase 3 trial is going to be managed by a new network, yet another one. This is the COVID-19 Prevention Network, or CoVPN. This is basically an NIH-run consortium being chaired by Larry Corey at University of Washington and Fred Hutch in Seattle, Kathy Newsel up in Baltimore at the Center for Vaccine Development at the University of Maryland. Basically what the NIH did is just they took all of their existing clinical trial networks, the HIV trial network, prevention network, the vaccine and treatment evaluation units, put them all together and have basically charged them with doing phase 3 trials. The plan is to enroll in 5 phase 3 trials by the middle of next year, a total of 30,000 participants in each trial. So that's quite a bit of people that are going to be vaccinated in the next year. These are the current sites. So this is the CoVPN sites in the U.S. There's 2 in D.C., including us and Whitman Walker. There are some international sites as well, mainly in South America and Africa. And the goals of the network really are harmonization. So harmonized protocol design for the phase 3 trials for all the ones at least that are going to be sponsored by BARDA and NIH, centralized laboratories that will do all of the immunology and virology testing, and a common DSMB as well. So really to ensure harmonization between all of these trials. Here at GW, the efforts are really building upon those that are already established by Mark Siegel and infectious diseases, and Mani Magnus, who's the PI of the clinical research site that's part of the HPTN network, NIH funded. So because of their strong work up till now in terms of HIV prevention trials, we're going to build upon that, but it's really going to be all hands on deck. All of the ID division is going to be involved, and then some, as well as our partners at the School of Public Health. Where we're at is for the Moderna mRNA vaccine is for prelim results from the phase 1 that I just shared with you. The plan for the phase 2 is to have the first blinded safety results from after the first dose of vaccine in the phase 2 by the first week of July. And based on that, we're going to start vaccinations in the phase 3. So the plan is to initiate vaccinations on July 9th, which, according to the timeline, will be before we have the results, the safety results at least, from post-vaccination 2 of the phase 2. But the plan is that the results from the safety results from after all the vaccinations are complete in the phase 2 will occur before initiation of the second vaccinations in the phase 3 trial. So very, very tightly sequenced. And this has been accepted by FDA. So the phase 3, which we're going to be a site for here at GW, in addition to the 72 sites I showed you, there's going to be a few more in the US. This is only going to be sites in the US for this first phase 3 trial. We're going to enroll 500 here at GW over the age of 18. The plan is we're being directed to enroll at least 25, but not more than 40% over the age of 65 or under the age of 65 and at risk. And that's someone with one of 5 different comorbidities that place them at higher risk of disease. Randomized placebo-controlled, observer blind, testing the 100 microgram dose of vaccine versus platelet saline placebo in a 1-to-1 randomization. Doses will be given intramuscularly, and it's 2 doses at 0 and 1 month. Just wanted to point out the demographics for who we're going to enroll. This is what was, this is from the study, the first 45 volunteers that were enrolled in that phase 1 of the vaccine in Seattle. So just to point out that they were predominantly white, 90%, which this is done in Seattle, albeit. But from everything that's been presented over the past few months, we all know that hospitalization and mortality is definitely disproportionate among the African American and Hispanic communities. And that is reflected in what we see locally. This is from the DC COVID website, DC government COVID website. So we are not going to repeat what was seen in Seattle. We are definitely going to do our best to make sure that African Americans and Hispanics are well represented in our at least local study population in the phase 3 trial. The endpoint is disease, symptomatic COVID infection. So this is an endpoint-driven study. So it is positivity by PCR plus 2 or more of fever, chills, myalgia, headaches, or throat, loss of smell or taste, or 1 or more these 3 respiratory findings. This is the timeline of the study. So 2 injections at day 1 and 29. The first planned interim analysis is going to be based on how many target cases are seen. So once 35% of the target cases are seen, which is estimated to have, if current infection rates continue, will be in about 3 months after all vaccinations are complete. If it looks like effective at that point, they will end the study. Or sorry, I should say they will unblind the study. The second interim analysis is planned after 70% of target cases are seen or occurred. The final target case number is 151. So all of the sample size evaluations are based on a target sample size, or target number of cases of 151. This is based on an assumed incidence in the placebo arm of 0.75% per 6 months, 90% power to detect a vaccine efficacy of 60%. What this really means is that this is powered to detect efficacy with a lower 95% confidence limit on the vaccine efficacy above 30%. I hope there will be a little bit of time for a few questions, because I'm sure there will be some based on our plans for the phase 3 trial of the Moderna mRNA vaccine. Just to say that there are other mRNA vaccines that are coming online. This just shows one. This is a slight change or a difference in the actual construct. It's still an mRNA vaccine, but it's what's referred to as a self-amplifying mRNA vaccine. This is being developed by Imperial College in London. They've just started phase 1 trial of this construct. So basically, the spike protein gene of interest is fused to a non-structural protein mRNA from, for example, an alphavirus. And this particular non-structural protein encodes for an RNA-dependent mRNA-dependent RNA polymerase, so that once it's the initial transcription in the cell, that RNA polymerase can make more copies of the spike protein mRNA. So you get more of the spike protein being produced, leading to a longer kind of duration of the vaccine protein in the body. So hopefully a better immune response. So just because of the time, I think I will not say anything more other than that this is now just this past week started a phase 1 clinical trial in the UK. I did want to mention before that time is up the second phase 3 trial that is being planned for the US under the CoVPN network, and that's the Oxford vaccine being taken up by AstraZeneca. I'm sure you've heard a lot of this about this vaccine in the news. They've just definitely been very good at promoting it. The investigators, that is, but it is one of the ones being supported by BARDA and NIAID. This is a replication-deficient chimp adenovirus. The reason for choosing a chimpanzee adenovirus is because humans should not have pre-existing immunity to it, so it shouldn't interfere with the immune response. That does express a full-length spike protein. So this vector is actually the CHAD vector has been around for a long time, and there's been lots of other infections for which, organisms for which vaccines have been developed to try to prevent. It's been used, lots of different infections, constructs have been tested, even in phase 1 and 2 trials, but none of which have progressed to licensure, mainly because they were shown not to work. So I think there's a lot of skepticism about whether or not this is actually going to lead to an effective vaccine. I think it remains to be seen. I'll show you in the next slide some results from an NHP study. It's well into clinical development, clinical trials in the UK, and it is anticipated that this will be the next phase 3 that will be started in the US, starting as early as late July, but most likely in August. We will not be a site for this vaccine. So this is the phase the phase 1/2 trial has already been started in the UK. Total of about just over 1,000 participants have been vaccinated already. We don't have any results yet. I assume that it's not unsafe, because we probably would have heard about this already. Last participant was vaccinated in mid-May. Hopefully we'll have results from this soon. This is the non-human primate study that has been published, at least preliminarily, that did show some binding antibody responses in the vaccinated monkeys and neutralizing antibodies as well. But if you look at the titers here, this is also based on the plaque reduction neutralization assay, just like the Moderna vaccine I showed earlier. They're about 5% of the level of titers that were seen in the human trial. Albeit this is a monkey trial, and you can't automatically extend results from that to humans, but not, from my read on this, not really rip-roaring neutralizing antibody responses. And then they also looked at, they challenged these non-human primates with virus after they had been vaccinated. They were able to detect virus by PCR in 2 out of 6 of the vaccinated monkeys versus 3 out of 3 of the controls. 100% of all vaccinated animals had detectable virus in nasal swabs. They said that they thought this was not unexpected because they had to deliver the challenge nasally. So that's possibly true. But then when they looked at lung pathology, they also were able to detect virus in the lungs of 2 out of 6 versus 2 out of the the 3 controls. So what's next, at least in the US? I just talked about the Moderna and AZ vaccines. The next 3 are still in preclinical development, but hopefully will be in clinical trials. All 3 of them, at least in phase 1 by the end of the year. So just to end off, those of you who know me know that I've been involved in developing a human challenge model for hookworm to test vaccine efficacy. There are some people who think that we should be doing this for SARS-CoV-2. I think from an ethical standpoint, there's too much that's unknown about this virus, and that even in low risk people, we potentially could induce severe disease, and no one would want that to happen in a challenge trial. So that is not yet being done, but people are advocating for it to speed up assessment of efficacy. I think I will skip over what I was going to say there, just to say that in addition to vaccines, that's not the only way to prevent infections, that monoclonal antibodies are also being looked at, both for treatment and prophylaxis. These are 2 of the companies that are kind of first out of the starter's block, Lilly and Regeneron. These are monoclonal antibodies targeting the receptor binding domain of the spike protein. They're the first one going in clinical trials, at least in the US, is a cocktail of 2 monoclonals from Regeneron that target non-overlapping sites of the spike protein, and that have been shown to neutralize SARS-CoV-2 virus. That really didn't show up well. We have started here at GW, just within the past week, an ambulatory study, so looking at COVID positive patients and giving them an infusion. It has to be delivered intravenously of this MAB cocktail to see if it modulates the infection. And Mark Siegel is PI on an inpatient study for the same cocktail. The CoVPN network is looking at using these the monoclonal antibodies, both from Lilly and Regeneron, for prophylaxis, for prevention, and these will also probably start in the near future, potentially as early as summer of this year, targeting nursing home residents and healthcare workers. And I will stop there.
David, thanks. That was terrific, and I think it's just what everybody wanted to hear is where we stand and how long this is going to take. There's the concern, at least for a while, that the disease was on the way down, at least in this area, make it harder for a vaccine trial to be completed since it's dependent on people becoming infected rather than, giving patients the virus?
I actually think that if we start seeing more cases here, it's going to make vaccine development, or the phase 3 trial at least, quicker, or we'll get it resolved quicker, because it's really, obviously, if too many people have been infected, we're not going to be able to vaccinate uninfected people. We're not, incidentally, we're not doing serology, we're not checking for infection before vaccinating. So obviously that might interfere with the vaccine trial, but my sense is that if we see the rate of infection going up locally, that it actually will make the time to determining efficacy even quicker.
The people that may be positive should randomize out, but looking for more infections in the area, I guess.
Yeah, that will be taken into account in the analysis, and that's already been built into the sample size calculations.
Right. We better watch how we word that phrasing of rooting for more infections, though, I think. So Dr. Gallo from Baltimore is suggesting that the polio vaccine will give people immunity to the COVID. And do you have any response to that?
Yeah, not just the polio vaccine. There's been a lot of conjecture and some epi data about BCG vaccines. So countries that have been, I think actually Dr. Axelrod presented on that a while ago. That some countries that have, and also oral polio vaccine, that regularly vaccinate or use that vaccine seem to have less severe disease. I think and it's thought to be a non-specific inflammatory response that might help with protection. I think that remains to be seen. That's just hypothetical at this point.
Do we have enough data now over the last 4 or 5 months to suggest that this virus is not going to be a problem with mutating and
like the flu does? Yeah, well, evidence to date is that at least the spike protein, there has not been a lot of mutation. If that remains to be the case over the next couple of years, then I don't think it's going to be a problem like we see with influenza. And another question was that, it seems like a lot of the new vaccines and even some of the older vaccines, like Zoster, we're giving 2 injections in a short period of time. But yet with flu, we've continued just to give 1 yearly injection. Is the trend changing in vaccine development now? Well, I think that has to do with pre-existing immunity. So, when the first time a kid gets vaccinated with flu, you have to give 2 injections. And then it's subsequent to that annually. And I think it's related to previous vaccinations and previous infection as well. The issue now, so that it could be that for SARS-CoV-2, the first, in a completely naive population, you have to give an initial 2 vaccinations. But if we do need to give boosters annually, or hopefully that won't be the case, but if we do need to do it, hopefully we just get away with giving 1. Do you have any advanced knowledge about the flu vaccine for this year, whether, what's in it? And I guess we don't have any idea about efficacy yet. No, we should know. I don't know the answer to that. I should, but usually the decision is made in February or March as to what the components are going to be. Okay. David, thanks so much for doing this. We're going to have you come back probably in the fall to give us an update, and hopefully it's a really positive update. But the final thing I'm going to ask you is, what's your best guess if this study was positive, that a vaccine would be available nationally?
No pressure.
This isn't a congressional testimony, is it?
Don't forget.
I think it's possible if everything goes perfectly and the Moderna vaccine works, that we could have large, widespread vaccinations as early as mid next year.
Wow. That still seems like a long way off. David, thanks so much for doing this. This is great. Everybody, we will have a grand rounds next week. It may be somewhat abbreviated, but we'll talk more about COVID. What else next week? See you then. Thank you.